This only tells probability. It doesn't tell what's actually going to happen. It tells probability. All 4 of these, that's 100% chance, will have a tail. So, the probability of them having a child without a tail is none of these. So that would be, the answer would be 0%. Okay? And what I want you to understand is anytime I am homozygous dominant for a parent, the big T is always going to show up in every single one of these. And so it is impossible for this allele to ever become homozygous and show up. So anytime I'm homozygous for the dominant trait, the answer is always going to be 0 for those that don't have a tail, or for those that would, whatever that trait is, that would be recessive. Okay? So that's the first practice. Okay, let's go to the next one. That says 2 parents are carriers for a deadly disease. What is the probability that they will have a child with this disease? Go ahead and stop now and try it, and then I'll go through it with you. Okay, thanks for doing that. Now what we're going to do is I'm going to go through this. Now the key word here is carrier. Carrier means automatically that they're carrying the trait that they don't show themselves in their phenotype, but they got the gene for it. They're carrying it on for the next generation. It might show up in their children, it might not, but they're carrying it for that next generation, possibly. All right? They don't have it, but they can carry it. So it automatically means they are heterozygous. So the trait, if we go back to the steps, the trait is the deadly disease, and the alleles are they have it or they don't have it. All right? The genotypes of the parents, we know they're both heterozygous, and now we're going to create gametes and analyze our Punnett squares. So let's do this. I can choose whatever letters I want to choose. I'm going to go with big L because they're easy to draw, equals no disease because they're carriers. They don't have the disease, they're carriers, and they're heterozygous, so no disease. All right, and little l would represent they have the deadly disease. All right, we'll just say dis for the disease. So now both parents are carriers. That means both parents are big L, little l. So if I were to determine what the gametes could possibly be, Dad could give a big L or a little l. Mom could also give a big L or a little l. And the way to show that in a Punnett square is, if I draw this box, cross, big L, little l for Dad. I'll put a big L and a little l. Mom's the same thing, big L, little l. Now remember what this represents. These are possible sperm. These are possible eggs. Now when I put them together, if that sperm and that egg come together, this is big L, that is homozygous dominant for no disease. This one box right here will be big L. We always capitalize the first one, and then the little l. Sorry. That is heterozygous for the dominant trait of no disease. This one right here would be big L, little l, also heterozygous for no disease. And then this one, something interesting happens here. I get little l, little l. That's homozygous for the recessive trait. The only way it shows up is if it's homozygous for the disease. So there's only one chance in 4, 1 out of 4. And the way I do this is I think of it as quarters. That's 25 cents. If I had 2 of them, it'd be 50 cents. 3 of them would be 75 cents. The 4th one would be $1. All right? That's 25%, 50%, 75%, 100%. If none of them, it's 0%. What is the probability that they will have a child with the disease? The only one that has the disease would be homozygous recessive. It's 1 out of 4. So the right answer is 25%. All right, let's do one more. it says, you're gonna try this. Why don't you do it, and then we'll work it out. Okay, go ahead and pause, and then we'll work it out. All right, thanks for that. Now let's give this a shot. Tall is dominant to short in pea plants. This is what Gregor Mendel found. He says a tall plant. So the trait is tallness in pea plants. The alleles are, if I were to draw this, big T, because we know it's dominant, is tall. Little t, which we know would be recessive then, is, don't do that, short. Okay? So it says tall is dominant to short, so that tells us our alleles. And it says a tall plant is bred with a short plant, and you get short offspring. What must be true of the genotype of the tall plant? This one's tricky. This one is not like the others. They want you to figure out the genotype of one of the parents. One question on the test, on the common assessment like this. It's a multiple choice, but you have to be able to do this. Work it backwards. So let me show you how to do this. To be a short plant, the only way the short plant can be short is homozygous, little t, little t. All right? Because if there's a big T there, it's tall. It's dominant. So I have a short plant. Now the tall goes with the short, and I get short offspring. I can get short offspring. So let's look at the possibilities for tall. It could be big T, or it could be big T, little t. So what we have to do is we have to check. We have to work it backwards. Now if I have a short plant, little t, and I breed it with that possible parent, big T, big T. If we go back to our first question, you'll see that anytime, remember, if you have homozygous for the dominant allele, that we have 100% chance that the dominant phenotype, which is what it looks like, phenotype means what it looks like, will show up. All four of these are going to be heterozygous for tall. They're all tall. If I got a short plant, short offspring, there's a 0% chance of that. That can't be my answer. However, let's check the other one. Big T, little t. Cross with little t. This one is big T, little t. That would be tall. This one's big T, little t. Big T, little t. That would be tall. This one is little t, so I have a chance for short there. And this one's little t, so I have another chance for short. The odds of getting a short one is actually 25, 50, 50%. Here it's 0%. So the only way I could have short offspring with my tall and short plant is if the genotype of my tall plant must be this. And if we use the right words, the right terms, that is heterozygous. Heterozygous for tall. That must be the genotype, the genes of the tall plant. Okay? The phenotype is it's tall. The genotype is big T, little t. It's asking for the genotype. In order to get short offspring, it has to be heterozygous. Okay. Final thoughts. If I have more than one trait going on at a time, because really when I do this, if you ever want to go into a career for this and you want to look at this, you're never doing one trait at a time. When we did our simulation in class, we had 15 traits going on at a time. Sometimes you're looking at hundreds or thousands of traits going on at a time, and we want to know probability. Well, that's fine, but we can still do what we've done here Even when you get to college, even when you get to grad school, you can still do this. Check this out. If I do big L, little l, crossed with big L, little l, the only chance of getting the little l allele to show up is here. That is 1 out of 4, or 25%. Now, if I want to look at another trait like big T, little t, so little t's, there's also a 1 out of 4 chance that the little t shows up. Now, if you remember, we did a lab in class where we flipped coins and we rolled dice, and I told you about probability. And the only reason I'm introducing this to you now is it's one of your math core content standards, so you'll be doing this in math at the end of the year. What are the odds that I get both big little l, and little t at the same time? It's 1 out of 4 here, 1 out of 4 here. To get them at the same time, I multiply them together, and I get 1 out of 16. If I have 3 traits going on, and I got another one that's 1/4, I would take 1/4 times 1/4 times 1/4. Let's say I want to change this, and instead of knowing the little t here, I want to know what the odds are of getting the big T trait and the little l trait. Then it would be 3 out of 4, so it would be 1/4 times 3/4. I just take whatever my odds are in each one, and if I want to know what it's like to have them together, what the odds are, I multiply them together. You won't have to do this in 7th grade, in genetics, in science. You will have to do it in math later on, so I thought I would go ahead and introduce it now so that you'll have an idea. Whatever the odds are on any one thing, if I want to know several things and I wanted to know them at once, I do the odds of each one and then I multiply them together. I hope that helps. I hope you're ready for this test. We're going to work more problems in class. I may actually give you something to take home over the weekend to look at, but you've worked 2 different SpongeBob worksheets. We've done a complete simulation. We've done some problems like with the Amazing Maze with the corn in class, although that happened very quickly. We've done a lot of this, and a lot of you are really good at it, and you've been really good at it from the beginning. I'll give you more practice over the weekend, and if you need my help, you can come in and see me, but this is it. This is the crux of the test. Knowing how to create Punnett squares and solving them, or reading ones that have already been made and seeing telling me what they mean. So thank you very much for watching. I hope this makes sense. This is 7th grade genetics, but if you have this down, you'll be able to do really well in high school when you get up there. Have a great day, and I will see you on Friday.